Hello everyone. Today I'm here to talk to you about the field of the science called epigenetics. Did you hear of epigenetics? Okay, so for those of you who didn't hear about it, epigenetics is a new and existing field. This video will give you a brief idea about what this rapidly growing area is. But before that, I would like to talk to you about the DNA Day. And every year in April 25th, uh, we celebrate DNA Day. So what is DNA Day? DNA recall of the two events in the history that contribute to our understanding about DNA. The story of DNA began on April 25th, 1953, where doctors James Watson and Francis Crick determined the structure of DNA. 50 years later, in April 2003, the Human Genome Project discovered the entire sequence of the human DNA. Research scientists are using the knowledge and technology generated by this project to further understand how your DNA sequence can contribute to disease. But how exactly are scientific made these discoveries? Well, it all began with a question or an idea that no one knows the answer to. This question or hypothesis can be thought of like a single puzzle piece. You have no idea how many pieces are in the puzzle, what the final puzzle is going to look like. You know the general subject of the puzzle, like for instance, but you want to know about it. So you start to think about it to help you find the other pieces that fit into the puzzle. It is just like you are doing an experiment. So you do one experiment to get a second piece, and you need to repeat that experiment. Every experiment you do has to repeat multiple times. You might add a reagent in a different order uh, one day and get completely different results. Therefore, it is crucial that experiments are repeated so that you can be uh, sure that your result on your puzzle piece is actually the piece that you think it is. After spending years collecting your puzzle pieces, you have finally put together a couple of them and you think you have a really interesting discoveries. So you present your discovery to a group of scientific experts in the field at a conference. The expert raise questions about how you put your puzzle pieces together, and the expert give you a lot of good ideas or mention things that you may not have thought of it before. So you go back to lab and do a few more experiments to address the question that other expert brought up at the conference. Now with some more experiments completed and replicated, you will feel even more confident about your discovery. So you move on the next step, publishing the result of the whole world to see. To do that, you go back to your results and write an article that describe your discovery and exactly what experiment you did to make the discovery. Then you send the report to a scientific journal where they have other experts in your field review or review report. If the experts agree with the law, how you fit your puzzle pieces, they agree to publish the paper. But if they don't think your pieces fit exactly, they will reject your work and you will have to go back and do more experiment to better fit the pieces together. So most scientists' works and discoveries are rejected multiple times until they eventually get published for the general public. So all of the scientific discovery that you hear about in news have gone through a rigorous review process and are only published if a large group of experts agree that the information is correct. But it still doesn't end there. Scientific discoveries are further tested when other researchers build new hypotheses of existing information and discover a new set of puzzle pieces. The The new puzzle pieces will only fit both the new and existing information are correct. A group puzzle pieces fit together to form a scientific theory. This process is constantly cycling where both new and old information are being tested and make it possible for a community of scientists to be certain of the theory that they generate. Now, let's see what is DNA. DNA is the most basic molecule building block of human being. Your DNA actually is your physical appearance, but it also play a role in determining your susceptibility to disease and even your personality. Everyone is a unique individual, and part of what makes individual unique is your distinct variation of DNA. DNA actually is made of the nucleotides that are the four small molecules: adenine, thymine, cytosine, and guanine, abbreviated by A, T, C, and G. The DNA in your cell is actually composed of two molecules of DNA held together by uh, nucleotides pairing with each other. The two molecules are said to be complementary. The shape that they form when paired is a double helix. It is the structure that Watson and Crick defined back in 1953, but now the nucleus pair together. DNA and nucleus pair up in a consistent way. Adenine always pairs with a thymine, and cytosine always pairs with guanine. A pair of nucleotides in a DNA double helix is called the base pair. Now, when we look at the double helix, we only need to know the sequence of one of the molecules. Why is this? So nucleotides pairing follows strict rule. We can always figure out the sequence of the complementary DNA molecule. Let's unwrap this DNA and take a closer look. If we unwind one of the molecules and write out its nucleotides in order, we can now read the DNA sequence. Here is the DNA sequence based on what we know about DNA-based pairing. We can go back to figure out the sequence of the complementary strain of the DNA. This is a sequence of the very beginning of the chromosome 1. This is showing the first 954 nucleotides. If you try to put the entire human sequence into a PowerPoint presentation, it would take about 6.3 million slides. The genome can be divided into coding and non-coding region. Of our entire genome, only 2% contain coding DNA. Genes are found in the coding region of the DNA. Genes are the region of DNA containing instruction of the cell to make functional RNA or protein. So what is protein? Protein is a large molecule that does a specific task in the cell, like breaking down cell waste or importing nutrients. It is believed that the human genome contains about 20,000 genes within the coding region of the DNA. So more recent information suggests that this number could increase to 40,000 genes when you take into consideration all coding genes and functional non-coding genes. The whole genome is a cookbook full of the recipe for the cell. Our 4 nucleotides are like the letters that make the word of the recipe. Each gene made up of the nucleotides is like a single recipe in the book. If you think about it, only 4 different types of nucleotides doesn't seem like it would give you a much variety, but think of how much the order matters in the letter in a book. One last thing I want to remind you of this, that if a gene is activated in a process known as a transcription, we say the gene is expressed. To carry out on the cookbook analog, this would be like the recipe or gene actually being made. This expression process is very important in the topic of epigenetics. Now, that you learn about what DNA does, we can talk about something we don't always think about it, how big it is. All of your DNA is stored within a compartment in a cell known as a nucleus. So the question is, what is the width of the nucleus in a human cell? The answer is, the nucleus is a tiny 6 micrometer. That is only 1/10 the width of the human hair. So the next question is, if you stretched all the DNA in your chromosome out and put them end to end, what would be the length of the DNA within the nucleus? The answer is 1.8 meters. To get a better picture of the what, putting 1.8 meters of the DNA into a nucleus, that's 1/10 as a width of human hair. You can picture trying to fit 10,000 miles of a spot into a regulation size basketball. Here is what the DNA inside of our nucleus looks like. It is pretty amazing to see of how much of there is floating around in a nucleus of each one of the cells. Now tell you to do an activity to see what our DNA looks like. Okay. Now I'm going to ask you to do an experiment to see what your DNA looks like. So start by adding a saliva to a tube you have. As soon as you do that, add 1-2 drops of the soap to the tube and mix it up. Then add a little bit of the salt and a few drops of the ethanol. Use a toothpicks to spool the DNA in the tube. Now that you have this DNA, can you tell the difference between your DNA and your parents? It should not. All our DNA looks the same from the point of view. There are differences we cannot see encoded in all your genome. So I told you what is the size of nucleus was and how long your DNA is. It seems impossible to fit all the DNA in such a small space. So how the cell accomplish this task? The cell does this by wrapping all the DNA around a protein known as histones. Histones are shaped like an actin. They have a circular core and they have tails kind of like tentacles that just stick out from the core. When DNA wrap around the set of the histone protein, the structure called a nucleosome. This is similar to taking a long street and winging to tightly around the spool to minimize its size. This nucleosome or spool of a thread can be wound even more tightly on the top of themselves to really tightly pack the DNA in this compact state. You can no longer see all of the DNA. Similarly, this tight packing prevents the cell from seeing some of the DNA. So DNA is wrapped around the nucleosome. What do you think would happen if the cell tried to get to some of the contact DNA? The answer is the cell cannot reach the DNA that is buried in the tightly compact nucleosome. Just like the genetic code that tells our cell what gene to read, we also have an epigenetic code that tells our cell which gene to read and when. Nucleosomes are super important in this process. The nucleosomes are marked with a different signal that tell your cell when to read which genes. In fact, you can think of nucleosomes like being traffic light for the cell. When nucleosome get a repressive mark, it's like a red light to tell the cell do not read or make this gene. The nucleosome can also be like a yellow light. It is ready to quickly turn red or green. Finally, nucleosome can have actively marked, which is like green light. It tells the cells to go and start transcribing this gene. Now that we have talked about and seen your DNA, let's talk about what protein in our cell interact with our DNA. As I mentioned earlier, our DNA needs to be expressed. The recipe needs to be made, and the transcription protein help carry out that function. An example of would be RNA polymerase, the protein that copies the DNA sequence into RNA sequence in the process called transcription. If our DNA get damaged, we have protein that will help to repair the DNA at those sites of damage. Without this protein, our DNA would be full of many more mutations. Now we know that DNA has to be replicated in order for a cell to divide, and there are proteins that will carry out the process as well. Without this protein, we would not be able to grow at all. These are important processes in the cell. And with these proteins, we wouldn't be able to exist. When we talk about the epigenetic, there are 4 proteins that are important to remember. First, writer protein. These writer protein mark on our DNA or histone protein. Second, erasure protein. These protein are marked on our DNA or histone protein. Third, the reader protein. This protein bind to the marker. The reader protein record other protein that can activate expression of the gene. Now, you know what DNA is and what the protein can interact with it. Let's talk about epigenetic. Epigenetic is a mechanism that change gene expression that doesn't involve change in the underlying DNA sequence. If we think about all the DNA in our cell like being that big cookbook, we talk about it earlier, epigenetic is like using highlighter to mark certain recipe. It doesn't change any of the letters, but it does change how the person might read that cookbook. Just like a baker might mark the recipe they use most, these highlighters make marks tell the cell where to look and their own huge set of the DNA. So we know there are proteins that write, read, and erase epigenetic mark. But how does these change gene expression? Well, there are 2 main mechanisms. The first is histone acetylation, and the second is DNA methylation. Concepts in order to do well in the game we are about to play. Remember these activist-shaped protein called histones from earlier. Well, small chemical groups called acetyl group can be added to the tail of these histone by the writer protein. We talk about it. This process is now as a histone acetylation, and it is an extremely important epigenetic process. This collection of the histone atoms is called an acetyl group. By adding this small group, the expression of the entire gene can be changed. The process of adding these acetyl groups is known as acetylation. At a chemical level, the acetylation physically loosens the DNA from the histone. This process is similar to how magnet ray repel each other. The reader protein can also come in and recognize acetyl marks. They bind and bring along other protein that express the DNA. The most important thing remember is that acetylation is like giving the cell a green light to turn on expression of the gene. As an example, in the DNA, the most important thing to remember is acetylation, like giving the cell a green light on the expression of the green cell. As an example, the histone on the top has no acetylation. That means their DNA is not accessible when a reader protein is nearby. It doesn't find any acetyl mark and therefore it doesn't bind. The bottom histone have acetylation, so their DNA is accessible and a reader protein will bind. This allows the reader to bring the protein that causes gene expression, now the sRNA polymerase, ultimately so acetylation. The second epigenetic mechanisms we are going to talk about it is DNA methylation. This small collection of the atoms is called a methyl group. Methyl groups are added directly to the cytosine nucleotides in a DNA, and this process blocks gene expression. This is known as a DNA methylation. This is different from the adding acetyl groups because the methyl groups are added to the nucleotides themselves. Do not histone protein, also the methyl group block the DNA expression. So how does this process work when DNA is unmethylated? The DNA is clear and it can be bound by a protein that promotes expression. This turns the gene on. When DNA is methylated, it's block the protein from binding and prevent expression. You can think of this protein sliding across the DNA like you might slide down a hill. If you hit a bump at the beginning of your ride, similar to these methyl marks, you might fall off the sled. Similarly, the methylated DNA bump off and prevent the protein that promotes expression from binding. This turn on the gene off. You can think of these methyl marks as being red stop, right? Light that tell the gene or cell do not to express the gene. Just remember, methylation must mute the gene. So we already know that all of us have a genomes. Well, did you know we also all have epigenomes? Our epigenomes is the combination of a histone acylation and DNA methylation throughout our entire genome, and it's unique from anyone else. If we didn't have epigenomes, our cell wouldn't differentiate and we would end up looking like this sad and differentiated blob here. One thing about epigenetic is that our environment and lifestyle can impact our epigenome. Think like our diet or use of the drug can change the marks. We have talked about it today. This is turn change how our genes are expressed and ultimately how we develop. You know any identical twins? Do you think they are identical in every way? Of course not. There are a slight difference in their appearance and personality. This may be due to the fact that although their genomes are identical, their genes are expressed differently. Their epigenomes are different. Does the environment affect? The answer yes, because the environment can change the outcome of the someone's epigenome. Now let's go over some existing research study that have uncovered some of these environment epigenome interactions. One study that showed that childhood experiences can have an effect on the epigenome in the later lives focused on the uh, maternal care in a rat. Rat typically lick their young in the first week of the life, an activity associated with motherly nutrients. Some rats participate in the publicing, and these pups grow up to be less and anxious and generally happier. Some rat moms ignore their pups, and their pups grow up to be more anxious and not natural their own young. Investigator at the McGill University showed that maternal care causes demethylation of the glucotide receptor, resulting in a mice who have higher levels of the glucocorticoid receptor in the brain. This means that these mice are less anxious. But is that necessary better? Rats living in the cities probably need to be more cautious in order to deal with the dangers like rat traps. One example of epigenetic happens in the bees. In each hive, there is one queen bee and a bunch of the drones that maintain the hive. Look at how different the queen looks from the workers around here. The worker bees and queen bees have very similar genomes. The differences comes from differences in their diet. Queen bees are fed royal jelly from a very young age. This royal jelly slimes genes called DNMT3. DNMT3 typically adds methylation or slimes queen genes in the worker bee. When DNMT3 is repressed and the queen bee is allowed the queen gene to be expressed. Epigenetic modification can also be inherited. One example of transgeneration epigenetic inheritance involves these agouti mice. Normally, these mice have a yellow coat and tend to be a bit like the mouse on the left. At the Duke University in 2003, researchers found that when they feed these mice floated supplements during a pregnancy, which helped to place DNA methylation marks, the mice would give birth to thin mice with burned coats. They found that the supplement promoted methylation at the site near the gene for coat color in the baby mouse genome. The brown baby mice had less of the risk of diabetes, cancer, and obesity than the yellow mice, which is interesting. Another example is how many of you have seen the recent news about an identical twins going to the space? Scott and Mark Kelly are identical 53 years old twins. During a study conducted over 2016 and 2017 looked at the congenitive physiological, physical, and genetic effect of the Scott spending a year in space while his twin Mark remained on Earth. What do you think they saw? Did the twin DNA actually change? No, the DNA didn't change, but the epigenetic mark did. Natural, a famous scientific publisher, recently released this statement to correct a lot of these inaccurate news article. Even a news stories on the Today Show had incorrectly reported the result of the study. So Scott DNA doesn't change, and actually not different from his twins. Instead, the expression of epigenetic code is changed. The expression of thousands of the gene, many related to scientific cold strays, was altered during Scott flight, including fluctuation in epigenetic age. Over 90% of the genes with the altered expressions return to normal post-flight, but a subset of the several hundred genes remained disrupted after Scott returned to Earth. These genes were returned space gene, an area of interest for future research overall. What these results mean is the yet to be determined. The sample size is one, but it is an interesting example of what type of research is being done in the field of epigenetics now that you have the tools and understanding of studying many epigenetic modification. Thank you, and I wish you enjoy the video.